Hello and welcome to the last in our series of anniversary lecture events to celebrate the 75 years of Great Ormond Street Institute of Child Health. Today we've got the future with two speakers who will introduce you soon, but we want to first really bring home about this new YouTube channel that we have, and for you to subscribe to our channel and help support all of our public engagement events at Great Ormond Street Institute of Child Health. And now I'll hand over to Professor Rosalind Smith and come back to you at the end for our question and answer session. Thank you.
So a very warm welcome to everybody to this, our third and final series of lectures in this lecture series to celebrate our 75th anniversary. So we've progressed from the past through to the present, and now we're looking at the future. We've got two fantastic speakers today, Julien Baruteau and Alice Giustacchini. I was telling Alice that I had to go out and find an Italian speaker to help me pronounce her surname. It did not take very long. So I hope I've got that more or less correct. So the usual reminder is to log into the Slido, www.slido.com, and to use the event code, which is #ICH75 future. And then you can ask a question or endorse a question that's been asked by somebody else. And there's also a poll so that we can get a sort of insight into the makeup of our audience today. So we've had a really fantastic series of lectures with Tim Cole, first of all with his long and distinguished career, the last 22 years of which were spent at the Institute of Child Health, followed by Manju Kurian and David Long, who are both very well established and distinguished PIs who have been at this institute for more than 10 years in each case. And now we have our final two, which is Julien and Alice, both of whom have come to ICH more recently. And you will, as Claire has said, see the previous talks and indeed the talks today on our YouTube channel. So let me introduce you to Julien. He is an MRC Clinician Scientist Fellow at ICH and an Honorary Consultant in Metabolic Medicine at Great Ormond Street Hospital. He graduated from Toulouse and specialised in the field of inherited metabolic diseases in Paris before moving to Great Ormond Street in 2012. He was always interested in doing research and undertook a PhD with our close collaborator, Simon Waddington from the Institute of Women's Health, completing that in 2017. He then undertook some postdoctoral research with Professor Paul Gissen at this institute, looking at translating gene therapy for inherited metabolic conditions. And in 2020, he was awarded the MRC Clinician Scientist Fellowship to explore cerebral arginine metabolism. Julien's work is within the Genetics and Genomic Medicine department, and his research group focuses on developing novel therapies for inherited metabolic diseases, particularly urea cycle defects. So welcome, Julien, and we look forward to your lecture, When Little Paws Take Big Steps.
Thank you very much, Rose, for this kind introduction. This is very much a pleasure and a privilege to be able to present at this 75th anniversary lecture series, presenting about the future of our institute. In the next 20 minutes, I will present some of my research work, and this title, When Little Paws Take Big Steps, is very much to illustrate the importance of animal research in my work and how this has enabled some key achievements in the last years. First of all, a little bit about myself and my background. So as you can hear it, I'm French. I performed my medical studies in Dijon, in Burgundy, in the center east of France. in a region with a lot of vineyards that I very much encourage you to visit. I then did my pediatric training at the Children's Hospital in Toulouse at a time where it was the first flight of the giant plane, the A380 in Airbus. And I decided to train in a specific field of medicine called inherited metabolic diseases at Robert Daubé Hospital in Paris, and joined back in Toulouse a couple of years. And in 2012, I decided to jump across the channel and to join the metabolic team at Great Ormond Street Hospital as a clinical fellow. And at that point, I met a lot of very enthusiastic clinician scientists, and I realized how much research was going on just next door of the hospital at Guistacchini. And I promised myself that I will join this institute one day. My journey at UCL started with an Action Medical Research Training Fellowship, which enabled me to undertake a PhD in gene therapy under the amazing supervision of Professor Simon Waddington at the Institute for Women's Health. And I then joined Guistacchini for a very inspirational project led by Professor Paul Gissen about translating this gene therapy into a clinical trial for rare disease. I started my research independence with the MRC Catalyst Fellowship, which was very much of a springboard to enable me to build up a strong application for a more prestigious fellowship, an MRC Clinician Scientist Fellowship, which started just at the time of the pandemic. Let me now introduce you a little bit more about the field of medicine I'm specialized in, inherited metabolic diseases. This schematic represents the metabolism in general. So that means all the biochemical reactions and pathways which enable a cell to work and life to happen. And if we simplify it, we can think that a pathway is very much like a chain. And if a link in that chain doesn't work appropriately, this will have an impact on the whole chain, will cause dysfunction of the cell, potential symptoms, and potential genetic disease. And at Great Ormond Street Hospital, there is a very special legacy about this field of medicine, because the person who described this specialty was Sir Archibald Garrott, who was a physician at the Hospital for Sick Children situated at Great Ormond Street in London in the early 1900s. And what he discovered is one of these conditions called alkaptonuria, which cause darkening of urine of patients caused by the accumulation of toxic metabolites. And this was published in this book, Inborn Errors of Metabolism, in 1908. And this field was for some time called Garrodian medicine. And this is very much meeting medicine, biochemistry, and genetics. Over the years, and especially during the second years of the 20th century, the amount of knowledge regarding this condition has exponentially increased. And you can see here the increase of the number of volumes of textbook regarding this specialty. Most of this, all of these inherited metabolic diseases, in fact, are rare diseases, meaning that they have, for each single disease, there are very few patients described, but because there are so many rare diseases, more than 7,000 described, that means that it represents a lot of people. More than 3 million people in the UK, more than 30 million people in Europe, more than 300 million people in the world. And if we would group all these people affected by rare diseases in a single country, this would represent the third most populous country in the world, meaning the United States. And we are very interested in these rare diseases at ICH because most of them are affecting children, especially young children, and most of them can cause severe diseases, with 1 in 3 children affected not reaching his 5th birthday. So if we go back to the metabolism in general, I have a specific interest in a pathway called the urea cycle that you can see here. And this pathway is very important because this can enable the detoxification of a highly neurotoxic compound called ammonia, which is synthesized by the breakdown of proteins. And this cycle enables the detoxification and synthesis of urea instead of ammonia to detoxify nitrogen. If this cycle doesn't work properly, ammonia levels will rise, and this can cause some acute neurological symptoms and can require acute admission in intensive care units. And you can see here, for example, a child which has required a blood filtration to restore some life-compatible levels of ammonia. If these patients are not treated quickly enough, this can cause some long-term severe neurological sequelae. You can see on the left panel a brain MRI with a severe cerebral atrophy, and on the right panel a pathological section of a brain with cystic formation within the hemisphere. So what is the standard of care for these urea cycle diseases? The main treatment is a protein-restricted diet. We bring just enough protein for the patients to to grow properly and to have a good protein turnover, but not too much to avoid the formation of too much ammonia. That means that the food with a high protein content is banned from the diet. That means meat, fish, eggs, for example, and the protein intake relies essentially on vegetables and fruits, which are weighted every day to ensure we don't get too much protein in the diet. This is combined with pills that we need to take multiple times a day to decrease ammonia levels. And if this combination doesn't work properly, we can perform a liver transplantation, which will cure the disease, but which will require lifelong immunosuppression and associated morbidities with the liver transplantation. In this group of urea cycle diseases, I'm interested in a specific condition called argininosuccinate lyase deficiency, or ASL deficiency for short. And this enzyme, ASL, is unique in mammals because this is the only enzyme enabling the synthesis of arginine, which is an important amino acid for the synthesis of various downstream products, especially nitric oxide, which is a messenger for various organs, especially the brain. So if this enzyme doesn't work, that means that arginine and nitric oxide levels will be decreased, and upstream the block, toxic like ammonia will increase. This disease was described for the first time 60 years ago in Manchester, and now we know that this happened in around 10 patients born in the UK per year. When I started my PhD, I performed a natural history study of that condition. And the main finding of this work was to realize that all these patients have a very high rate of neurological complications, developmental delay, learning difficulties, epilepsy. But strangely enough, the patients who have had a very good control of ammonia levels since birth because of early diagnosis do present the same rate of neurological complication. That means that high ammonia levels is not the only cause of the neurological insult in this condition. I have expanded this work with a natural history study, which is looking at all the other urea cycle conditions. And we are recruiting patients all across the country. And this has shown us that these diseases are really severe, causing hospital admission more than once a year for these patients, and true hyperammonemic episodes, usually around once every 2 years. And the age when these patients are developing high ammonia levels is essentially during early childhood, when these children are contracting intercurrent illnesses, or when they are teenagers. The disease I was mentioning, ASL deficiency, has a good mouse model which recapitulates well the human disease. And as you can see on this picture, the affected mouse has a shorter size, has abnormal fur compared to a normal animal, has a reduced survival with most of the animals dying at around 3 weeks of age, and high ammonia levels in the blood as expected. With the help of Claire Duff, with a postdoc in the lab, I'm working on understanding the role of ASL deficiency low arginine and low nitric oxide in the brain, and how this impacts memory. For that, I'm using two different models. One is a novel mouse model in which I'm studying behavior, looking at energy, metabolism using different tools, and looking at differential gene expression at a single cell level in the brain. We're as well using another model, which are skin cells, which are modified using biochemical scissors, CRISPR tools, and which are differentiated into neurons with the help of some specific chemicals to look at their electric activity, electrophysiology, and differential gene expression. This is an example of a test looking at testing memory in mice. This is called the Barnes maze. We have, for example, a circular platform with 20 holes all around, and one hole which is linked to an escape room. And there is a training phase during the mouse will learn where the escape room is. And on a test day, this escape room will be removed. And I will look at how long this tested animal will spend in the target quadrants during a 2 minutes period to see if the animal remembers where the escape room was. And you can see that this differentiates really well between an animal with preserved memory, which will spend quite a lot of time in the target quadrant here at the top of the picture, and an animal with impaired memory, which will look around and try to find the escape room all along. Another aspect of my work is to develop alternative to organ transplantation using gene therapy. So for that, this is quite simple. When we have a mutated gene, which is causing the synthesis of a deficient protein, the purpose is to add a therapeutic gene using a specific vector, which will then enable the restoration of a competent protein within the target cell. And for that, which is really important, is to look at to choose the right vector. And for that, we are using some modified viruses, because through evolution, viruses have been able to get a very good capacity in penetrating target cells. So this is, for example, a work I did some years ago using an adeno-associated viral vector, an AAV virus, which had been modified with the insertion of a therapeutic gene for ASL deficiency. And when I performed a single intravenous injection in an adult mouse and was looking at this animal over 1 year, I was able to restore survival in the treated animal in blue versus untreated animals in red. I was also able to normalize ammonia levels over time, here in blue again versus untreated animals in red. And the aspect of the animals treated in blue versus untreated showed a good restoration of the fur aspect and a bigger size as well. And this was published some years ago. So the limitation of this AAV vector is the fact that the therapeutic gene brought with this vector is not transmitted during cell division to the daughter cells. That means that if we inject a neonatal mouse with the rapid liver growth, the therapeutic gene will not be brought forward over time, and then we will lose some efficacy of this approach. So this is an example, for example, of this. When you inject some AAV vector with a reporter gene in adult versus neonatal mouse targeting the liver, you will have a very rapid decrease in terms of protein expression after neonatal administration. So for that, I'm developing, with the help of Lucia Turamanidou, who is a PhD student in the lab, a novel approach with an integrating gene therapy, a vector which is called a lentiviral vector. And with that, when we have performed some experiments injecting with a single intravenous administration a neonatal mouse, and looking at this animal over 3 months, we were able to restore very well the survival, again, in treated in purple versus untreated animal in red, in normalizing ammonia levels in treated in blue versus untreated animals in red. And after 3 months, it was very difficult to differentiate mutants having been treated versus a wild type animal. I have as well some interest in another specific project, which is called the HORACE clinical trial. This is a project which is led by Professor Paul Gissen in collaboration with Ian Alexander at the Children's Medical Research Institute in Sydney. And this is designing a clinical trial for another urea cycle disease called ornithine transcarbamylase or OTC deficiency. This is a project which has been ongoing for some time now, with efficacy studies performed in mice, safety studies performed in monkeys. We have now finished to work on the regulated the paperwork and we'll be able to submit clinical trial authorization next month. And we hope being able to start this clinical trial and inject patients next year. This is using some AAV vectors, but with a different AAV, which has a more potent potency in penetrating liver cells. And this should enable a much prolonged and much better protection against high ammonia levels for these children. Another aspect is to develop novel assays to assess the efficacy of the new therapeutics we are developing in the lab. This is an example of an assay that has been developed with the help of Simon Eaton, who is a principal investigator at GOS ICH, with the help of Danny Perrocho, who is a research associate, and Sonam Gorum, who is a postdoc in the lab. And the principle of that is to use what we call stable isotope. We inject some labeled metabolites, which will go through the cycle, and we will measure the amount of labeled urea at the end. And if we have a deficiency through the cycle, the amount of labeled urea produced will be much reduced, and this will be able to tell us about the functionality of this pathway in real time. So this is a work that has been developed in human cells, which has been validated in mice, and which now being translated into humans. So why working at Great Ormond Street Institute of Child Health is so special? To me, the main point is very much about the people I have met in this research institute, the amount of expertise, all the knowledge, all the know-how, and all the experience as well, especially in the field of gene therapy, which has been developed by many teams in many fields of medicine with very much success. There is as well access to a large network within the institute, within UCL, at a wider level as well internationally, access to cutting-edge equipment as well, which can enable us to design very successful, powerful experiments. Another aspect is the close link with the hospital and the access to large cohorts of patients affected by rare diseases and the teams looking after them. The two institutions are very much feeding themselves back and forth in developing new therapies or understanding better the mechanism of diseases and bringing new therapies or new diagnostic tools or new prognostic tools in the hospital. And what I find absolutely fantastic, very inspirational, is to see very much about the patient's focus approach on both sides, and everyone very much try to bring, to make a difference and to fulfill the unmet needs that the precious patients who are looking after have. How do I foresee the future of the field of inherited metabolic diseases? The story started more than 100 plus years ago, and many important steps have been reached throughout the years. But what I really find fascinating is the fact that at the moment, there are a huge number of clinical trials upcoming, many novel possibilities in terms of modification of gene therapy strategies, which are being developed, new small molecules, and for some diseases which didn't really have any therapeutic options some years ago, the patient really now has to choose between maybe 2 or 3 clinical trials. This is completely novel. And I very much do hope that in the future, all subpopulations of patients affected by this condition, especially the sicker ones like neonates and new children, will be able to benefit from these novel therapies. In the same way, I do hope that the whole spectrum of these inherited metabolic diseases will be able to benefit from these novel therapies, especially the ones for which there is currently very little of term of therapy, for example, mitochondrial diseases. With the development of more efficacious therapies, this is very much raising as well awareness about these rare diseases. And I really do hope that in the future, we'll be able to develop novel tools in order to diagnose these conditions very early at a stage where there is minimal symptoms. So for example, with generalized newborn screening. Another aspect to me is very much the optimization of the healthcare pathways to try as much as possible to reduce the delay to diagnosis and initiation of treatment if there is one. As a conclusion, I think that rare diseases provides very much a unique insight into understanding better the pathophysiology in general, and especially for more common diseases. The novel technology that we are developing in the lab offer new hope for rare diseases. And this is very much a heartfelt feeling. GOSH ICH really provides a unique environment for enabling transformative discoveries for our patients. I would like very much to acknowledge my group, Claire, Sonam, Danny, Lucia, Paul Gissen, who has been a very successful mentor for many years now, his group with which we are working really closely, our collaborators at ICH, at UCL, in the hospital, at national, international levels, my funders, and thank you for listening.
So, Julien, that was fantastic and very inspirational. And thank you so much for opening our ICH 75, the future, with that wonderful talk. So Julien has given you the link to the Slido. We're going to go straight into Alice's talk, but if you could put your questions for Julien into the Slido now, that would be great. So, and it's no accident that our two speakers today, looking to the future, are both from continental Europe. And it's a huge privilege that we have within our institute to be able to welcome the brightest and the best from across Europe. And we intend to do that going into the future and beyond. We celebrate the fantastic contributions from our European colleagues. So, and I want to move on to talk about Alice Giustacchini, who is Italian and obtained her PhD from San Raffaele University in Milan under the supervision of Professor Luigi Naldini, with a project focusing on the role of microRNAs in the regulation of hematopoietic stem cell functions. She then moved after her PhD to the UK to undertake a postdoc position in the laboratories of Adam Mead and Sten Erik Jacobsen at the University of Oxford. And during this postdoctoral attachment, she focused on the development of novel single cell approaches to resolve the impact of cell heterogeneity on leukemia therapy response. In 2019, she came to UCL within the Faculty of Life Sciences, and then was recruited here to a lecturer's position in infection, inflammation, and immunity, and also as a group leader. And Alice's group's research focuses on normal and malignant hematopoiesis, with a strong focus on stem cell biology, single cell technologies, and T-cell based immunotherapies. And essentially her group aims to understand what makes leukemia unresponsive to conventional therapies, and they combine single cell multiomics with immunological and stem cell functional assays to develop precision immunotherapy strategies. So welcome, Alice, and we're looking forward to your talk entitled Fighting Cancer, One Cell at a Time.
Thank you very much to Professor Smith for the kind introduction. I hope everyone can see my slides and hear me okay. So it's a great pleasure to be presenting as one of the speakers as part of the GOSH ICH 75th anniversary lecture series. And it's a particular honor to be one of the two speakers representing the future of this institute. So during the next 15 minutes or so, I'll talk about the exciting research that is going on in the lab at the moment. And in particular, I'll focus on how my research group is benefiting from being based at GOS-ICH. So just before I start, just a little bit about myself. As Professor Smith introduced, I'm Italian. I obtained my research degree at university in medical biotechnology at the San Raffaele University in Milano, where I also did my PhD in the laboratory of Professor Luigi Naldini. In 2013, I moved to the UK to undertake a research project in the laboratory of Professor Jacobsen and Mead at the University of Oxford. And in 2018, I moved to UCL, initially as a UCL Excellence Fellow. And since 2019, I've been a lecturer and a group leader here at the Great Ormond Street Institute of Child Health. And here I'm really building my research group. So you can see us here in this picture. We are based currently at the Zayed Center for Research into Rare Disease in Children. And we are a very enthusiastic and dynamic group and very thoughtful, as you can see in the picture. And during these few years, since I've been based at GOS-ICH, we've been successful in securing quite a few fundings for external sources from charities and funding agencies, including the Great Ormond Street Hospital Biomedical Research Center. So these funding are really enabling us to perform our research, but what is our research about? So since I did my PhD and been very fascinated by stem cells, and stem cells are very special cells. We have stem cells pretty much in any tissue in our body, and they carry very specific biological features, which I'm listing here in this slide. So stem cells are very long-lived. So to preserve their cells for a very long time in our organism, they tend to cycle, proliferate quite infrequently. And this characteristic is called quiescence. When stem cells do cycle, they can either make more of their cells, and this is called self-renewal property, or they can either differentiate to give rise to all the cell types that are part of the tissue they are stem cell for. And this property is called multipotency. So if we think about it, stem cells in a way are a little bit like children. When they are immature, they have a great potential to become pretty much anything, while when they start to grow, to differentiate and proliferate, they become more and more specialized toward a specific lineage and a cell type. And each of those cell types will carry out specific biological functions within the tissue. So the stem cells that we're interested in are the stem cell in the blood, and these are also some of the stem cells that have been characterized as first. This is due to the high accessibility of the blood as a tissue as opposed to other tissue in the organs, in the organism. So blood stem cells are also known as hematopoietic stem cells. They reside in the bone marrow, and they are capable to provide us with all the cells that we have in our peripheral blood long for our entire life. So from stem cells, all sorts of peripheral blood cells can originate, including cells that carry out very well-known functions, such as erythrocytes, indicated here, which are in charge of delivering oxygen to the tissues, or other immune cells, which are responsible for helping our organism fighting infections or cancer as well. So since blood stem cells are quite long-lived, what can happen during our life is that a mutation occurs in these cells. And a mutation is nothing but a mistake that happened, and it's then inserted in the genetic code. This mutation, unfortunately, in some cases can lead to aberrant function being acquired in these stem cells, and this can lead to their malignant transformation into blood cancer or leukemia. And the resulting leukemia cells can then expand in taking over the bone marrow and the peripheral blood, and they will really prevent the other cells in the blood from carrying out their function. So one interesting thing about some types of leukemia, in particular the leukemias of a myeloid origin, is that they preserve a hierarchical structure similar to that observed in the normal hematopoiesis. In this case, we have malignant stem cells at the top of the hierarchy, with more differentiated leukemia cells at the bottom of the hierarchy. So when we think back to the characteristics of stem cells that I presented at the beginning of my talk, quiescence, self-renewal, and multipotency, these characteristics are really unique and beneficial when observed in healthy blood stem cells. And they are really the basis for the success obtained with regenerative medicine, such as transplantation of hematopoietic stem cells in the bone marrow. But this same characteristic, unfortunately, when associated to malignant stem cells, they can really make these cells quite resistant to the treatments. And they are leading, therefore, to treatment failure, and they are often associated to disease relapse. And this is the hypothesis at the moment. So most of the chemotherapies that are used for treatment of leukemia and cancer, they really target the highly proliferative cells in the leukemia. But they don't or fail to effectively target the malignant stem cells, which, as I said before, are quiescent. So as a consequence, these malignant stem cells will be able to escape the effect of the drugs and they will be able to survive, leading to residual disease occurring in many of these patients. And this, therefore, can be followed by disease progression and relapse, unfortunately. So these malignant stem cells have really been the focus of so much research in the past few years, and they are quite critically important cells to understand the behavior of, to be able to target them clinically. So what we have been struggling in the past few years in the research field has been really to be able to characterize these malignant stem cells effectively. And the problem is that they are pretty much indistinguishable from their normal counterpart. And in some cases, in particular during residual disease, they're quite rare. So since we don't have a way to separate them out from the other cells in the blood or the bone marrow, we haven't really been able to characterize them as we wanted to. So what I did during my postdoc was to set up a technology by which we actually look at these cells one cell at a time. And by doing so, we can distinguish the malignant cells from the normal cells in the same patient, just based on the presence or the absence of a mutation that is associated to malignant transformation in that particular patient. And how are we actually managing to isolate single cell and study them? So this is what we call now a single cell technologies, and this is very fast growing in the field. There are now several approaches to isolate and characterize single cells. We can isolate them in individual wells of a plastic plate. We can capture them in nanochips. The example that I'm depicting here in the picture is actually showing how we can now also capture single cells in tiny droplet of oil. So here, these circular shapes that you see are oil droplet, and inside each droplet, we have one single cell captured. So this way, we can isolate one cell from each of the other cells present in the same tissue, and we can then characterize them one at a time and see and identify those cells carrying the mutations and separate them from those not carrying the mutations, so the healthy ones. And then we can zoom in and study these cells even more in details for what they are, for what is basically happening in the cell at the time of the analysis. So one particular analysis that we're interested in is called RNA sequencing. So we basically looked at the count and quantify the RNA molecules. So the RNA molecules are pretty much telling us which proteins are being expressed and produced in each individual cell, and proteins are the main players of cell biology. So this is really important for us to understand which proteins are being produced in a cell, so we can potentially find proteins that are specifically expressed in the malignant cells, but not in the healthy cells in the same patient. And this is really a powerful method to allow us to design therapeutic strategies that will be specific to the tumor without affecting the normal cells in our body. And here it looks how it looks like when a single cell analysis is plotted. So basically each symbol in this plot is a single cell, and these groups of cells that you can see are created are just the group created based on how similar some cells are, based on the expression of thousands of those RNA molecules that I talked to you about. And here, based on the color code, you can see we can distinguish cells carrying the mutations, so the leukemia cells, from those cells that are healthy in the same patient. And we also have cells isolated from healthy donors as control. So this is, of course, an overall picture of what single cell analysis can generate, but then we can zoom in specific RNA molecules, in specific encoding for specific proteins, and we can identify molecules, as shown here, the expression of which, indicated on the y-axis, is higher in the malignant cells in red, as opposed to the healthy counterparts. And this is really the ideal outlook that we would like to see for a therapeutic target, because it's very, very specific to malignant cells. And this we can only achieve with single cell technologies. So here at Guistacchini, we're particularly interested in studying pediatric myeloid leukemias, both chronic and acute. And acute myeloid leukemia, or AML, in particular represent an unmet clinical needs. These children are treated with intense chemotherapy, which can induce complete remission in a growing proportion of them, but is unfortunately associated to high rates of relapse. And once again, disease relapse in these children, as in adults, has been associated to malignant stem cells, which are present already at diagnosis, are not affected very much by chemotherapy, and they increase in number during relapse. And not only they increase in number, but they also increase in heterogeneity, meaning that they become very variable, very heterogeneous, pretty much like a moving target, very, very difficult to catch with traditional conventional chemotherapy. So the way we are trying to catch these malignant stem cells is once again with single cell technology. And you can see here, it's in this plot, each dot, once again, it's a single cell. What we are measuring here with the color coding is the potency of these cells. If you may remember, as I presented at the beginning of the talk, one particular feature of stem cells is their capacity to be multipotent. So here, this color gradient is measuring how potent each of these cells are. And we identify in the darker color the cells carrying the highest potential, so the putative leukemia stem cells, while gradually in the colors of blue and light blue, we have more differentiated leukemia cells. So in this way, we can identify targets and treatments against not only the more differentiated cells, but also the very rare therapy-resistant leukemia stem cells that we're really trying to eradicate. And we can do this in the diagnostic samples from patients. This is from a patient that presented at GOSH at diagnosis, but most importantly, we can also track and observe what changes in these malignant stem cells after chemotherapy. So not only we can find therapeutic strategies to eradicate them, but also potentially to prevent disease relapse. And how are we planning to do that? So how can we translate single cell sequencing, single cell technique to their clinical application to eradicate these very quiescent and dormant stem cells, as you can see depicted in the cartoon? So, and here I think it comes one of the biggest privilege of being based here at Guistacchini, because we have a great opportunity to work in close collaboration with research teams and clinical teams here at Guistacchini and GOSH. In particular, the groups of Professor Percy Samroli and Dr. Sara Gorashan, who lead the application of advanced cell therapies, which have been extremely successful for the treatment of other leukemia. And these advanced cell therapies are called chimeric antigen receptor T-cell therapy or CAR T-cell therapy. So how do this therapy works? Once we identify a target specific for the tumor cells with our single cell analysis that I showed you before, CAR T-cell therapy, then for CAR T-cell therapy, the next step is then to isolate T cells from the blood of the leukemic patients. So T cells are immune cells with a natural function of being able to mediate killing of other cells. So what this chimeric antigen receptor therapy does is to boost patients' own immune response. And this is achieved by introducing, using a lentiviral vector, novel chimeric receptor, chimeric antigen receptors, or CAR, which goes on the surface of the T cells isolated from the patient and are designed to recognize specifically the tumor molecule that has been identified by us in the single cell setting. So these cells, once they are infused back in the blood of the patients, they will be able to find the tumor cells and mediate their killing in a very specific and with high specificity. And most importantly, irrespectively of the quiescence of the cells. So this therapy can mediate the killing also of malignant stem cells as well as other leukemia cells. So once again, to wrap up this presentation, our research benefits greatly from being based at GOS-ICH. So we are developing in our lab novel techniques, but we are also given access to very advanced technique at the forefront of genomics, in particular through UCL Genomics, which is also based at the Zayed Center for Research, where our group is based. And this really puts us in the best possible position to discover and translate novel therapeutic targets to the clinical application. But not only, so we are not only going from the single cell to the bedside, but this is a bidirectional crosstalk because all the information are also fed back at our research. I didn't have a time to show this during the presentation, but we also contribute to the characterization of already ongoing and successful cell therapies ongoing at GOSH. So we have the opportunity to even better refine these ongoing treatments for this information to then be fed back to the clinic once again in a circular manner. And this is, of course, very much possible thanks to the great collaboration that we have in the Institute with Professor Sergi Castellano on the genomic side and Professor Persis Ramroglia and Dr. Sarah Gorashian on the clinical side. And of course, with a number of important collaboration nationally and internationally as well. And with this, I would like to conclude. Thanking you very much once again for the opportunity to present in this lecture series. I would like to thank my group, which is really the driving force of all the research we are doing. I would like to thank our funders and thanks everyone for the attention.
Thank you very much, Alice. That was really fantastic. Thank you. I think I'll hand over to Claire and to Dan for the questions.
Yeah, it was great. Thank you both. Really, really interesting talks. I just want to call out to the audience again. So you can still submit your questions via the Slido app. So that's hashtag ICH75Future. And also just a reminder to please subscribe to our channel. We've had a great response so far. We've got 98 subscribers. So please subscribe. There's a little button just down here somewhere and hopefully get us over the 100 mark by the end of today. So first question I'm going to ask is to Alice and Julien really, but Alice first. How's COVID affected your group? I mean, especially as a new group leader, how have you found that the pandemic has really, you know, that momentum you need to get a group going? How have you found that that's really affected you?
Yeah, that's a very good question also because the timing wasn't great. The COVID pandemic happened, I think it was the first year after our group started. So there was a very critical time point where everything was starting. So we did have some delays, of course, during the closure of the Institute for a few months. But I have to say it was also a way for us to find other way to communicate. And we managed to establish even a better connection that we had before in the end, which it's been very, very fruitful for when we came back to the lab. So we focused a lot on analysis and planning. So we were ready to go as soon as we got back to the labs.
Yeah, great. Lots of planning time. Julien, do you have anything to add to that, or how did you find the lockdown situation?
Yeah, I echo what Alice said. That was very much at the time when my fellowship was just starting, and having the building closure, the mouse colony to be reduced. That was a dramatic attrition of the whole work that was going on. But hopefully now things have moved forward in a right positive way. So we are in a new pace, and we try to go through it with success. So that's hopefully just some memories, nothing else.
Great, thanks. Okay, we're going to go over to Dan now, who's in the Institute. I think he's found someone to ask a question to you. Daniel,
hello,
you there?
everybody. Here I am in a microscope room in the spirit of trying to get as many scenes of ICH in as possible. I'm here with Rachele Rossi, who is here ready to ask her question. So Rachele, if you would like to fire away.
Yeah, I would like to ask, what do you enjoy the most of your work, of your job?
Julien, should we go to you first?
I think what I very much enjoyed here is the people who are working here. Very much the discussion we have, the collaboration we have, the amount of discussion we can get, and the insight from different people when we discuss about some our research, how we can improve, how we can think about in a different way, optimize our experiments. This is very much what I enjoy in that environment.
Okay, Alice?
Yeah, that's a good question. So what I enjoy the most is looking at results. So usually now, it's slightly a different job. So as a PI, I spend lots of time also writing grants, which I don't like as much. But really the exciting part of this work is when one experiment is performed and you have a result. It's really the time by which you try understand what happened, if it's gone as you thought it would go, or if it's gone another way, and then why, and that interpretation discussion. So that's really the fun part of a job.
Thank you.
Okay, thanks, Dan. We'll go back to the Slido now, and then we'll come back to Dan for another question in a minute. Here's one that came onto the Slido for Julien. It says, great talk, Julien. You've got lots of projects. How do you keep all those balls in the air while still maintaining a life outside of work? I mean, what's life? I don't know. But
well, that's a good question. I try to tend to get all the balls in the air at the same time. That's well, I think a lot is about relying on good people in the lab. That's I'm not alone. I have really a team working with me, and we have collaborators as well within ICH and within UCL, who will really help us to generate results. So that's very much a collaboration between people, and I think I'm lucky to have a good mentor, good environment, and a very resilient wife as well, which helps.
Yeah, great. I think, Roz, did you put your hand up?
I did, Claire. I didn't know what the protocol was.
Yeah,
I just wanted to say something that relates to the last couple of questions from a funder's perspective, because I chair an MRC panel, and I chair various other funding panels. And the funders are really, really concerned about the impact of COVID pandemic on biomedical research. And so there is usually most funders have put in a page for applicants to fill out to talk about what the impact has been. And please do take that seriously. It's not just the impact of lab closure and so on, but it's also the personal impact, people who've had to stop or reduce the amount that they can work because of caring responsibilities in particular. So I think the whole community really wants to kind of support you and get us through this difficult time. And so do make sure you fill that out carefully and put your hands up and ask for help, particularly whenever you're experiencing difficulties because of the pandemic.
Great, thanks, Ros. That's a nice, comforting statement for all of us starting new careers in this field. So, Alice, I've got a question for you on Slido. Which feels into what Ros is saying as well. What one piece of advice would you give to a PhD student at ICH?
Well, I guess it's that's a difficult question. You need to think back a little bit, a few years ago. I guess it's always look around yourself. So it's very easy when you get very much involved into your PhD in getting very focused in a narrow field and super specialized. I think this is really a time where you can take the opportunity to interact with other people doing different things and keeps your mind open, because it's still a time where, you know, you could still adapt or change field. So it's really a time of to be open to other things, not too specialized. So that's probably the suggestion that I would give.
Yeah, absorb as much information as you can.
Absolutely.
Yeah, great, thanks, Alice. Julien, a similar kind of question, but with a different sort of focus. What piece of advice might you give to people looking to become a group leader?
Well, that's a good question. I think what is important is to have a good questions, a good field of expertise in a way that you can fill in, to have a good network of collaborators. And I think a mentor we can help you to thrive in the right way, and to try to engage early in some opportunities you have to get exposed to writing articles, writing small grants, presenting your work. So all the skills which will be required at some point in your work at a later time point when you will be you will have more responsibility. So trying to diversify as much as you can your opportunities and gaining the skills early as well to get them ready. And speak to people to try to know the right path and not to miss the opportunities.
Yeah, networking is key. However you can do it, virtual or in person. It's great. So we're going to try and go back to Dan. I hope he's found somebody else to do a question. Dan, are you there?
I'm here. So yeah, now we're in a tissue culture room. So this is what a tissue culture hood looks like, just for anyone that's interested. But I've got Will here with me who'd like to ask a question. So Will, say hello and fire away.
Hi everyone. Could I just ask, given that you're both scientists from Europe, what was it that drew you to set your groups up in the UK? What made it the best destination for you to work here?
Okay, Alice, should we go to you first?
Yeah, well, many reasons actually. So of course, scientific excellence, that's the reason why I moved. In my particular case, I was really interested in a specific topic. I wanted to study malignant hematopoiesis, and I targeted the group that I wanted to group, and I went to that specific group. And that's absolutely scientific excellence. I have to say that now, for my stage of career at the moment, apart from scientific excellence, there's also access to many opportunities to progress, to gain your own funding, and to establish yourself as an independent researcher. So there's something you can't do anywhere, and here you have particularly good chances.
Yeah, I would echo what Alice is saying. That was very much about the opportunities to get to join a group with a huge of expertise, and having this proximity with the hospital, and to have a very dedicated person with a huge amount of experience and knowledge, and the opportunity to grow here with lots of different stream of fundings and availability to join a team with international recognition.
Great. Thank you both. So we've got a very technical question here now from Terence Stevenson on the Slido for Julien. Can Julien explain in simple terms to a non-cell biologist why a lentiviral vector passes genes to progeny, but adenovirus doesn't?
Yeah, I mean, this is like the lentiviral vector is derived from the HIV virus, so from retroviruses. That means that these viruses will synthesize a double strand of DNA, which will then be able to integrate directly within the chromosome. This is something that AAV don't do. These adenoviral AAV vectors, what they do, they will have your double-stranded DNA molecules, a circular one, what we call an episome, which will stay in the nucleus. But that's not a chromosome. That means that's during the cell division, that's not transmitted. So you are losing your therapeutic gene that way.
Fantastic. Thank you very much. I think we're going to have to wrap up the questions there. But that was really good. I think we probably just need a session of question and answers in the future. So, stay on to, subscribe to our channel. Actually, I've just checked, we're on 106. So yay, thank you, everyone. We made it over the 100 mark. I'll hand over to Ros just to wrap up the session. But thank you very much. It was very enjoyable.
So I think we've had a fantastic last hour with two wonderful future, well, already research leaders, but going on to much greater things for the future. So our future's in very good hands, I feel. And thank you so much, Julien and Alice. I also want to say a thank you to the other speakers in this series. So Tim Cole, Manju Kurian, and David Long. We've really had a fantastically successful 75th anniversary celebrations. I want to thank Phil Mason, who's not been visible in any of these lecture series, but has been extremely hardworking, extremely effective, and has supported all of the speakers and the organisers really superbly. And of course, I want to thank Claire and Dan, whose enthusiasm and energy has really made all of this happen, ably supported by Nicole Hoffmans. So huge thanks to you both, Claire and Dan, because without your experimentation and your great ideas, a lot of this fun stuff wouldn't have happened. So thanks to all of you. It's been a great set of lectures, and thanks to you all for your participation, and have a really happy Christmas.
Thank you.